Hi there, everyone. We are just going to let a few more folks trickle in, and then we will get started with today's webinar. Alrighty. Hello, everyone, and welcome to the Society for the Study of Reproduction webinar entitled Deciphering the Ovarian Microenvironment Across Developmental, Spatial, and Temporal Axis with Dr. Monica Laranda. My name is Jordan Shilley, and I am the webinar specialist for SSR. Also with us is Dr. Jane Fenelon, who is your moderator for today. If you have any questions or concerns about this webinar, please feel free to reach out to me directly at SSR headquarters via email at info@ssr.org. Just a couple of housekeeping items before we get started. All participants are muted with the exception of the speakers, moderator, and myself. The webinar is scheduled for 60 minutes and is being recorded. It will be posted on the SSR website shortly thereafter for members to access at their convenience. In just a few minutes, the moderators will begin with a brief introduction followed by the speaker presentation. Please feel free to submit your questions during the presentation using the Q&A text box on the bottom middle of your screen, and the moderator will read them to the speakers when the question portion of the webinar begins. And with that, I am going to kick it over to Monica and Jane, and we can begin.
Thank you. So as mentioned, my name is Dr. Jane Fenelon. I'm a member of the SSR Virtual Education Committee, and I'm very happy to have with us here Dr. Monica Laranda, who is one of our rising stars in reproduction. Monica completed her PhD at Northwestern University with Larry Jamison, and then went on to complete postgraduate training in the laboratories of Takashi Kurita and Teresa Woodruff. In 2016, she then became an investigator at the Stanley Mann Children's Research Institute and is an assistant professor in the Department of Pediatrics at the Feinberg School of Medicine at Northwestern University. She is also the Director of Research for the Fertility and Hormone Preservation and Restoration Program at Anne and Robert H. Lurie Children's Hospital of Chicago. We're very excited to hear what she's got to say, and take it away, Monica. Thank you.
Thank you so much, Jane. And thank you to the nomination and selection committees for this Rising Star webinar. It is truly an honor to be one of those chosen to be considered a rising star for this society that I've grown up in and really do value. So thank you so much. And thank you all of those of you who are attending. I have a few disclosures. One is that I'm an advisor to Dimension Inc., which is a 3D printing biotech company, but none of the research that I'm presenting today will involve any of their products. I'm also a section editor for Reproductive Biomedicine Online. I want to start with some of the motivation for my research and just provide some context. So at Lurie Children's, we see patients at increased risks for premature gonadal insufficiency. And this is really, an increased number of children survive cancer. So 7 out of 8 children will survive their cancer, but because of their treatments that allow for this survival of some of these horrible diseases, they may become infertile. And this remains a primary concern for some of these cancer survivors or survivors of certain treatments. And 75% of young adults who are childless at the time of diagnosis report a desire to be a parent in the future. And parents want fertility preservation options presented regardless of their infertility risk, prognosis, or prognosis of their child. And so some of the diagnoses where there is an increased risk for premature gonadal insufficiency would include some of this extracranial solid tumors, rhabdomyosarcoma, and other diseases like sickle cell anemia that use some of these chemotherapeutic agents. There's also genetic causes of premature gonadal insufficiency, and these are some of these are considered differences in sex development. And so that would be mixed gonadal dysgenesis or complete androgen insensitivity syndrome, for example. And so while we offer fertility preservation at Lurie Children's for those with ovaries and testes or genetic gonads under certain circumstances, my lab is interested in ovarian biology. And so some of the comorbidities that can occur with premature ovarian insufficiency is something that we consider. And so a person with an ovary who is born might have the most number of follicles that they will, or oocytes that could become potential eggs right around birth. And then it depletes over time naturally to a menopause around 50 years old. And with addition of these treatments, there could be a reduction in this ovarian reserve. And then induction of menopause early on. And when we think about menopause, we think about some of those comorbidities that may be due to the reduction in the systemic hormones. So the ovarian hormones that could help with memory or psychological well-being, promote cardiovascular health, bone, protect for gynecologic cancers, and induce good wound healing. Another important thing is that around the world, there have been several long-term studies that have demonstrated that the life expectancy can be reduced by 2 years because of some of these comorbidities. And so what we do is we offer ovarian tissue preservation for those with significant increased risks of developing premature ovarian failure through their treatment or disease. We perform unilateral oophorectomy because decades of research in animal models have demonstrated that the other ovary may compensate and maintain serum hormone levels. And the other ovary releases more eggs when stimulated with assisted reproductive technologies. Additionally, a study looking at over 20,000 women in Norway discovered that there is a little difference in age at menopause between normal and women with one ovary removed for a benign purpose. And so this is why we remove one whole ovary. And then we process this ovary into cortical strips and cryopreserve for future use. And currently the only option for restoring fertility after cryopreservation or ovarian tissue cryopreservation is autotransplantation of this ovarian tissue. And approximately 140 live births have been reported worldwide. The rate of autotransplant recipients that have restored fertility is between 20 and 40%. And the hormone production lasts for up to 12 years, but the average is between 2 and 5 years of what has been reported. However, there's a possibility of introducing cancer cells, and I'm showing an example here from work that I did in Teresa Woodruff's lab, showing that there's acute lymphoblastic leukemia cells or SALL4 positive cells within the cortical tissue, within that same piece that would have been saved for patients' future use. And of course, other people have shown a similar thing with several different techniques, but this tissue would not be transplanted back as is. And so another thing that we consider when our goal is to maximize the reproductive potential of that ovarian tissue is the age which the ovarian tissue is cryopreserved. And so you can see from this Oncofertility Consortium National Physicians Cooperative data that there has been a significant increase in OTC for girls within that consortium under 18 years old over time. And at Lurie Children's, our patients range in age from approximately 5 months to 23 years old with a median age of about 10 years old. And so these patients are within the age range of subfecundity, which means that they have fewer euploid eggs than the ideal reproductive age. And so there are both scientific and clinical purposes for engineering an ovary, and this is what we intend to do in our lab. And we can improve our understanding of the follicular microenvironment and how it influences quiescence growth and maturation by isolating follicles and testing them in this engineered environment. And so we could perform also xenograft analyses, and this can influence our clinical goals of creating a safe transplant that removes cancer cells, prevents premature primordial follicle activation, improves current rates of live births, and increases the longevity of hormone production. And so when we're considering what type of engineering component or microenvironment that we would want to support a follicle, we have to consider, of course, those support cells that surround it. And so the follicle is made of the centralized oocyte, as many of you know, that requires the support of those neighboring granulosa cells in order to survive and mature. And the use of alginate beads as an encapsulating material to support the physical structure of the ovarian follicle was a very critical advancement for in vitro analysis of follicular genesis. However, the alginate is not degradable and either needs to be modified or so that it can become degradable or removed to enable the last stages of growth and maturation. And so this was our motivation to have an open pore design. And we did this through 3D printing gelatin scaffolds to enable the three-dimensional follicle support, that sphere, to grow and mature and degrade that gelatin material. And so we tested several architectural designs. I'm showing the one that we chose. That was the intersecting 60-degree advancing angle. And you can see what a mirroring small secondary might feel based on this Z-depth analysis. And regardless of what type of scaffold we use, if the follicles were able to come in contact with multiple struts, so the 2 and 3 strut contacts, as shown here with the follicle in green and the oocyte in the center here, then the survival went up. And so we chose this design because it had more opportunities for the follicles to contact those struts. And then you can see it growing in culture. So this is a day 8 image with the oocyte in the center, the basement membrane on the outside, an expansion of those follicles. And so when we were first testing out these scaffolds, we showed the follicle dynamics in vitro. So the secondary follicle was seated within the 60-degree advancing angle, as cultured for 8 days, and HCG was added to the media. You can see cumulus cell expansion, and then there was a release of the egg through the microporous scaffold. And so then we thought that this might act as a great transplant, and we used green fluorescent protein tagged ovarian follicles and transplanted them into the mom that did not express GFP and whose ovaries were removed. So we put the bioprosthetic ovary, you can see it here within the ovarian bursa, and you could see the oviduct here that is not fluorescent green. And this mom was able to produce GFP pups from this 3D printed bioprosthetic ovary. And so I feel like I can't give this rising star presentation without discussing the real influence of my postdoctoral work and how it led to some of the research that I'm going to show from my lab now. And so this great experience at Northwestern University with Teresa Woodruff as my mentor, and being familiar with the Oncofertility Consortium, and then developing the bioprosthetic ovary and engineering designs after testing and analyzing many different materials led to the transition award by the Burroughs Wellcome Fund. And so I then in 2016 moved to the Stanley Manny Children's Research Institute at Ann and Robert H. Lurie Children's Hospital, which is still a part of Northwestern Medicine at Feinberg. So I didn't move far. But this gave me the opportunity to have a clinical insight into a lot of what's going on for fertility preservation. And this is really supported by some nurses and clinical research coordinators and surgical fellows, for example. And you can see here my co-director of the Fertility and Hormone Preservation or Restoration Program, Dr. Erin Rowell. And so because of this, we have access to human tissue from the pediatric population that I really didn't look at before. And so it became very interesting for me to not just think about what that ovarian environment is, like I described at the beginning of the talk, but what is the developmental, spatial, and temporal dynamics that happen within the ovarian tissue. And so I'll start with the developmental. And so you can see very starkly that these prepubertal ovaries, so 4.6 years old, are much smaller than the postpubertal ovary, even when this young woman is just 16.4 years. And this 11 times growth actually happens prior to any presentation of external Tanner signs or pubertal status changes. And so we know that there's a lot of different components that may be at play in addition to follicular genesis. And so this is what a postpubertal ovary at 13.8 years old, and then the 2.5-year-old prepubertal ovary looks like when we process it for ovarian tissue cryopreservation. And so we first bisect that ovary. You can see a large antral follicle coming out here. We remove the medulla, or sometimes we don't remove anything because it's within that thin or thickness where the cryoprotectant will penetrate as is. And then the medulla is scraped and then cut into strips. So while this is happening, these materials, tissue fragments and cumulus oocyte complexes are released into the media. And this is one of the opportunities that we've taken to gather research material, and approximately 91% of our patients actually donate their media to research. And so using those research materials, I'm showing here some of the tissue fragments. We started noticing that the prepubertal population had more oocytes or more follicles within some of those fragments than the postpubertal ovary. And that might seem kind of obvious to some people, but we were really interested as to why that is. And so you can see here that 45.5% of the prepubertal tissue fragments that were released during the OTC processing of those fragments contained follicles, or primordial follicles specifically, and then 10% of the postpubertal fragments contained follicles. And so this was after looking for pieces that had follicles by the method that was established by me and others, including Francesca Duncan in the Woodruff lab, to see those clearings on that tissue. And so after doing H&E tissue sections and counting those follicles per area, you could see that the total follicles per area were of course significantly different. And then the primordial follicles per area were also significantly different in the prepubertal tissue fragments. And so this made us think about, okay, within this process, how should we dissect out those tissues for OTC? And it became really interesting to us to look more closely at the anatomy. And so any tissue that comes out of the body during a surgery must have an ovarian, it must have a tissue biopsy sent to pathology. And so we're able to use those slides used for pathology for research as well. And so we scanned these H&E images of tissue biopsies, and you can see that there's anatomical changes in the ovary across the pubertal development. And here we're showing ages at the bottom. So you can see this ovary at the youngest time point that we're showing here is 2.69 years. There's the ovarian surface epithelium, and all of these are taken at the same depth apart, approximately 1.5 millimeters in depth. You can see that there's also the development of the tunica albuginea forming across puberty and thickening. And then you can also see here the cortical medullary junction forming between the cortex and the medulla, starting peripubertally at 9.85 years old. So according to this person's Tanner stage, she was still Tanner 1, so not yet going through having physical signs of puberty. And you can see that the cortical medullary junction persists post-pubertally. And this is actually how the ovarian tissue cryopreservation methods were developed, just saving that cortex where the primordial follicles or quiescent follicles reside. And so we asked, is there the presence of a cortical medullary junction? And of course, I think by these images, it's obvious that most of the pieces that we looked at, or most of the tissue biopsies that we looked at that had a clear orientation for these purposes did not have that cortical medullary junction, but most of the post-pubertal did. And when there was a cortical medullary junction, there seemed to be no difference in that depth as measured from the ovarian surface epithelium as to where that junction started. And then going back to the tunica albuginea that is also forming across puberty, we wanted to ask, is there a presence of tunica albuginea as defined by these dense cells just under the ovarian surface epithelium? And we could say yes in 5 out of the 11 for prepubertal, and yes for the majority of those postpubertal biopsies. And then how deep was that tunica albuginea? And it seems to be growing in the postpubertal ovary versus the prepubertal ovary and thickening across that transition. So then we also looked at overall follicle density down, I believe, to 3 millimeters in depth. And so you could see that there's much more primordial follicles within the prepubertal ovary, of course, and that the deepest primordial follicle is deeper in the prepubertal ovary than in the postpubertal ovary. So then the tissue seems to be thickening and transforming, and the cortex seems to be getting smaller as there's a depletion of primordial follicles within that region or spread across a larger area. So there's definitely still some more anatomical investigations to do. So now I want to talk a little bit more about spatial as I'm describing some of these things that happen across development. The ovary is divided into cortical and medullary compartments. So as I had mentioned, the cortex contains those primordial follicles, and this is the region that's saved for OTC. The medullary region also contains large growing follicles, and this region is removed and does not withstand the cryopreservation process and is very vascular. So when we performed decellularization, which is the removal of all of these, all of the cells and an enrichment of matrazone proteins and a lot of these fibrous proteins, you can see that the cortex has much more organized bundle formation and sheets and walls within that region. The medulla, of course, has these larger pores to accommodate those vessels, or that has been made by those vessels. and by those growing follicles. But we're also seeing additional flexible fibers and less organization and sheets making up those walls. And so we wanted to ask, are there different matrosome proteins in the cortex versus the medulla? And so in order to do this, we performed a proteomics analysis. And first we sliced a porcine ovary as a model organism in the axial and sagittal directions in 0.5 millimeter regular slices. We decellularized those slices again to enrich for those matrosome proteins. And we performed LC-MS and validated with immuno-PCR. And so we found over 1,000 proteins, but 317 were significantly different across slices. 82 were matrosome proteins that passed our stringency analysis, and 42 were significantly different across the slices. 11 of those matrosome proteins have previously not been identified in ovary protein analyses in any mammals. And so this is the breakdown. We found collagens, ECM glycoproteins, ECM regulators, ECM affiliated proteins, and proteoglycans, many of which were significantly differentially expressed across compartments. And because we did this in a regular way, we were able to develop SAS scripts with the help of Richard Leduc from Neil Kelleher's group at Northwestern, and be able to map those proteins across those sections, and so across the ovary. And so this represents approximately 1/8 of the ovary, and you can see that the collagen 1 is more highly expressed in the cortical region, or the 0.5 millimeter slice of the porcine ovary than in the medulla region. And so again, because we did this in those regular slices, we're able to investigate some proteins and how they compare with the standard deviation from the LS means in the cortex, which is the 0.5 millimeter slice through the medulla. We're also interested in mapping the physical properties across the ovary. And to do this, we used atomic force microscopy through nanoindentation. So for this model organism, we used a bovine ovary because it does appear more similar to a human ovary. And it has a clear cortex within this region here. And so we found that the ovary, this is a 1 millimeter slice, was much more rigid in the cortex than it was in the medulla region, significantly so. And so right now we're working on doing a high resolution type mapping at different regions of a similar slice, and then using a smaller probe, atomic force microscopy, and correlating these with features within the ovary. So primordial follicles, areas of just interstitial cells, vessels, etc. But from our preliminary data, we're able to average out these higher resolution maps and coming up with the same or very similar readouts as we did for the bulk rigidity testing. So it's promising. And so the Duncan lab has also used atomic force microscopy to define the rigidity of young versus old mouse ovaries. And they found that old ovaries were much more rigid. And so they also stained with picoceros red to identify the collagen content. And they also found that the old ovaries had much more collagen than the younger ovaries. And then when they treated it with collagenase, they were able to restore that same amount of rigidity between that's similar to the younger ovary using the old ovaries with the collagenase treatment. And so because we knew that this data, we wanted to confirm that the rigidity of the bovine ovary was from matresome proteins. And so we decellularized bovine ovaries, and you can see that here. We were able to make gels from the cortex. And so we tested the gels that were made from cortex or medulla regions with, again, atomic force microscopy. So we were looking for rigidity, and you could see that there's still a significant difference from the cortex, although much lower than the whole tissue, of course, but significantly different in the cortex from the medulla. And these are just, again, those matresome proteins. And so with this and other analyses that we did, the cortex tissue we found was 7.6 times more rigid than the medulla tissue. The cortex ECM is 1.1 times denser than the medulla ECM. The cortex ECM gel is 150 times more rigid than the medulla ECM gel. And so now I want to relate some of these findings and research studies to the temporal axis, which is across follicular genesis. And before I do that, I just wanted to describe that when we auto-transplant ovarian tissue that has been cryopreserved for future use, we see that there's an increased recruitment of primordial follicles or increased activation as identified by a spike in AMH production as shown here. And so this is Silber's work, but many others have seen very similar results, where after the transplant has been, after the ovarian tissue has been transplanted, the FSH levels drop, an indication of resumed function, and then an increased spike of AMH happens and then decreases. This has also been shown from studies in Israel and Austria that have xenographed human tissue, human ovarian tissue into mice. And so you can see that there's an increase of growing follicles within 3 days, an increase in growing follicles and decrease of primordial follicles within 3 days or over the course of several weeks. And so depletion of primordial follicles can reduce the longevity of an ovarian tissue or engineered transplant because there's really only a few directions that those oocytes can go. And so it's either being activated to grow and then developing to atresia, or developing and ovulating out an egg. And so with this increased activation, there's a decrease in the reserve, a decrease in function and graft longevity. And so I'm showing this SEM again because we wanted to ask the question, can we control follicular genesis through physical and biochemical cues? And this is something that my lab is very interested in. And so since we've mapped those proteomics, using proteomics, we've mapped the matresome proteins, and using AFM, we've mapped the rigidity. We wondered if the ECM itself can have both of those biochemical and physical cues. And there's reasons to think that it can, because the matresome has been shown to have tissue-specific properties. So this was a paper that was published while I was a postdoc in Teresa's lab, where the decellularized bovine ovary was again made into this DECM ink, or decellularized ECM ink. It was fabricated with a small percentage of PLGA, and this process had gone through, the tissues had gone through this same process for heart, kidney, liver, muscle, and uterus, in addition to the ovary. And you can see that when human mesenchymal stem cells were grown on these materials, there was a difference in affinities to adhere, to survive, and to proliferate that was different between each tissue. So another way to understand how physical cues can control follicular genesis, again using that alginate bead, with increased percentages and therefore rigidities. And the 3% alginate you can see here resulted in an increased ratio of androstenedione and progesterone to estradiol within these murine follicles. And so you can see that there was a smaller follicle than the ones that were in a smaller percentage alginate or less rigid alginate. And this actually changed some steroidogenic production. And Nagamatsu and others identified an increase in murine primordial follicle activation when they used collagenase and trypsin. And so this increase in activation was actually reduced back to the normal levels by just adding hyperbaric pressure. And so this shows that physical cues can also affect primordial follicle activation or transcriptional control. And so one way we might consider identifying or testing how the different physical properties might influence follicular genesis is again using these alginate percentages. And while the rigidity or the stiffness of these gels have been tested previously, I think it's important to use the same type of methodology in order to compare where these rigidities might fall within or in comparison to the live tissue. And so we reanalyzed these alginate at different percentages and with different crosslinkers to see if we could achieve some of these higher modulus percentages that exist within the cortex. And so another thought that we had is, of course, this is live tissue that has all of these cells and other components in it. And is that exactly what the follicle is feeling? But recently, Uni and others showed at a much smaller scale that they are identifying similar ranges of rigidity. You can see here by kilopascal within the ovarian tissue, again, with these an atomic force microscopy type of nano indentation methodology, but at a much smaller scale to get a topographical map. And so it seems that even at these smaller scales, the follicles might be feeling some of these more rigid environments. And so another thing to consider is, are these matrixome protein changes consistent across species? Because this consistency across species or evolutionary conservation might imply importance. And so we looked at some of our favorite matrixome proteins, collagen-1, amylin, agrin, fibronectin, vitronectin, and collagen-4, and found that this a very similar ratio or difference across the cortex versus medulla concentrations existed within the bovine ovary as what was found in the proteomics analysis. And so some of these proteins are of interest because in particular, amylin and agrin are extracellular glycoprotein or a large proteoglycan, which might be involved in signaling mechanisms. And so this is also present within the human ovary as shown by immunofluorescence around primordial follicles. And so again, looking back at the standard deviation from LS means maps and looking at the cortical region as identified by the 0.5 millimeter slice, you can use collagen-1, which is this known matrixome protein of the cortex, to identify and localize that amylin-1 is more highly expressed in the cortex. Again, this is the porcine proteomics analysis. And so it might be controlling through a TGF beta mechanism or through integrin binding primordial follicle activation at the granulosa cells. And this is something that we're actively pursuing. And so we're developing some of those materials within our lab to do so. And so how we're doing that is taking bovine ovaries, performing decellularization, and then sorting out the protein of interest, in this case, amylin or agrin. So using magnetic activated protein filtration steps. And you can see that when we do this from our decellularized ECM materials, that we can maintain collagen 1 relatively similar across all of the magnetic passes, but we can reduce the amylin 1 concentration within that material, or the agrin concentration within that material. And then when we make these gels from our depleted proteins of interest, they actually stay at the same rigidity as the cortex gel, as designated by this dotted line. And so the next steps that we'll perform in the lab will be culturing those follicles within gels or 3D printed matrices. So we're able to print these extracellular matrix materials using the fresh techniques. So this is a suspension bath. And this is an example with a collagen 1 ink. This is what the cortex DECM inks looks like, or the medulla. Again, performed with that 60-degree advancing angle. And so we're able to make these specialized inks from the decellularized material. And again, we wanted to see what that Young's modulus, or the rigidity of these gels, looked like. Cortex, of course, is higher than just collagen alone, even though most of it is collagen, and it's significantly higher than the medulla. But there doesn't seem to be much change within the depleted gels that were 3D printed when you remove amylin 1 or agrin. And so to wrap this up, in summary, I hope that I described that the pediatric ovary is fundamentally different from the adult ovary. And this brought us or got us interested in the developmental axis of the ovary, and investigating how that the anatomy changes and how that might influence oocytes that are then produced into eggs within the ovarian tissue that we cryopreserve in our pediatric patients for future use. And that ovarian tissue engineering aims to expand safety and efficacy of cryopreserved ovarian tissue and improve birth rates and transplant longevity, which is a long-term goal. That 3D printed bioprosthetic ovaries scaffolds can restore fertility and hormone function in mice, and that an ideal engineered ovary will consider the microenvironments, the different components that I presented to promote appropriate survival, growth, maturation of quality eggs. And there are significant differences in the composition and abundance of some matrazome proteins between the cortex and medulla, that the bovine ovarian cortex is more rigid than the medulla, and that the ovarian decellularized matrix can be made into these materials that we hope to then explore more molecular mechanisms and properties of follicular genesis with these materials that we've made. And so with that, I would like to acknowledge my lab members, the fertility and hormone preservation and restoration team, my collaborators, of course, the participants who have donated to research within our program. We owe a lot to them, and they really inspire a lot of our research. And of course, the funding sources. Thank you so much.
Thank you, Monica. That was a really fascinating talk, and congratulations on being a worthy recipient of the Rising Star Award.
Thank you.
If you'd like to stop sharing your screen, I think we can do that. Sure. And we'll take any questions you may have. We've got 2 already in the Q&A, but please feel free to add more. So the first question comes from Camilla Bridges, which asks, which said, and she's asked, I once read that ovaries actually shrink post-menopause. Is this due to the loss of follicles, or do you believe there is another reason behind this?
That's a great question. I guess I haven't witnessed what the size looks like after for a whole postmenopausal ovary, in the same way that we can see, a whole prepubertal and then postpubertal and then reproductive adult ovary. But if it does shrink, I mean, the ovary itself is very dynamic during, the cycle, and so those antral follicles can expand and make the ovary seem larger at different points in the cycle. So we would also have to take that into consideration, but definitely follicle dynamics contribute a lot to the size. But we think, because we've looked at the H&E sections from the prepubertal and postpubertal ovaries, that there might be some other things, including vessel infiltration, which might regress in postmenopause. I don't know. That might be really interesting to look at.
Related to that, is it the loss of antral follicles that makes the menopausal ovaries more rigid then, do you think? Because they're not being sort of pressed against, if that makes sense?
Well, from Francesca Duncan's data, it seems to be that there's more fibrosis that is occurring, and I think that's probably known. In addition to what she showed with those murine ovaries with increased collagen deposition, but I'm sure that she could answer that a little better than I can. But it seems to be that there's more rigidity because of the collagen content and other inflammatory type responses.
Yeah. Camilla also asks, and this is sort of related as well, does the expression of the matresome proteins change over time as the ovaries age?
That is something that I would love to test in our system. I know that Omorum's group, the Uni's, the first author, had investigated some matresome proteins by immunofluorescence in prepubertal reproductive age and then postmenopausal ovary cortex. So they didn't look at compartmentalization or overall abundance in the way that we did with our proteomics analysis. But there is some indication that it does seem to change over time.
And we have another question from Pei Zhao, who's saying, super cool work. Is there any molecule in the follicle responsible for sensing the rigidity of the surrounding tissues that we know of?
Yeah, so there's some mechanotransduction pathways that we're particularly interested. I think there's been several groups looking at the Hippo pathway, which I think is the most well-known mechanotransductive pathway. But there's definitely ways that granulosa cells or theca cells might be able to respond to changes in their extracellular matrix environment by, so the extracellular matrix proteins can present different paracrine factors, they can present different binding sites that then could get transmitted to the follicle itself. So I wouldn't say that there's one signal or one factor that would be influenced. It's multiple things that could affect the follicle. And a lot of this is done with human mesenchymal stem cell work in the engineering field, and looking at how to differentiate other types of stem cells, for example, by changing and modifying the morphology or the pressure of different cells.
Yeah, because I was really fascinated to see with your 60-degree angle one that the follicle doesn't actually need to be completely surrounded. It just needs to have some pressure from some sides. And
yeah, it's really interesting. And so when we developed that, I started looking more into some of the biomaterials journals, and they had it seemed to have been known that aggregates of cells could maintain their round shape overall, but then have these other cells kind of anchor them within square or sharp kind of angled triangular shapes. And it was pretty interesting.
Yeah, it's fascinating. We have another question from Shuzhao. Great talk, Monica. What is the primary driver to make follicles move from the cortex to the medulla region during their developmental process? Do we think it's a mechanical or a biological factor, or both?
This is one thing that we discuss in our lab a lot. Like how can we see, first of all, if follicles are moving? So are follicles moving, or are other cells filling in around them? That's something we don't know, and would love to find out, because I don't know, stromal cells might migrate around to fill in spaces, so then they kind of get pushed in more into the medulla. If anyone knows that answer, I would love to start there and then develop some experiments on figuring that out. Yeah,
because everyone thinks it's the oocytes, but it might not actually be them. It's probably the surrounding stuff. Erica is asking, great talk, Monica. I may have missed this, but does the rigidity of the cortex contribute anything to blocking activation of the primordial follicles? Or possibly, does the primordial follicle move to the medulla to be activated? I suppose that's related to the previous question as well.
Yeah, so it seems like the follicle is activated from multiple signals that are from the oocyte that seems to be known, and mechanisms that seem to be known, but also from granulosa cells. Kate Hardy's work showed that there's an active a proliferation activation pathway that might be downstream of TGF beta to promote that initial squamous cell to dividing granulosa cell that then becomes cuboidal and actually starts that activation process with the follicle going into the next stage, which would then be zip transitional, primary, secondary, etc. So I guess to answer your question, there's a lot of different signals that might come from either the oocyte or the granulosa cell to start that activation. And then what was the second part of the question? Oh, the rigidity. Does it prevent?
Well, does the primordial follicle move to the medulla to be activated?
Yeah, and I think the question was also, is there a rigidity component that prevents
contributing to blocking activation? Yes, because when you were doing your transplants, it seemed there was a big increase in primordial follicle activation. So
right, so PCOS ovaries have been shown through magnetic resonance elastography to be more rigid than normal cycling ovaries. So there might be a component with maybe not activation, but actually continued growth and ovulation because of the extra rigidity that exists within the PCOS ovary. And previously, drilling has been used for PCOS patients or anovulatory patients to induce activation. We also know that fragmenting the ovarian tissue can induce activation. But I don't know if we can add pressure to prevent primordial follicle activation. I'm not sure that has been shown in the lab yet, but it's something that we're interested in.
Is that where your scaffold could help if you transplant it with the scaffold?
Yeah, that's why we've been testing the different rigidities and noticed that that like even when Teresa's group, Jessica Hornick, had isolated out primordial and primary follicles and grew them together in a more rigid bead, it was still much less than the rigidity that we had measured exists within the ovary. And so the point of those experiments were to get them to grow and to always get to that egg. But now when we're thinking about that transplant longevity or prolonged hormone function, this is now something that we're more interested in. Can we control that activation?
Yeah. John Epic is asking, are changes in the matrosome characteristics a cause or a result of changes in follicular dynamics, do you think?
I think it's both. So I think this also goes back to one of the other questions about follicles potentially moving through the different compartments. There's a lot of, matrix metalloproteases that are, we know are secreted by the follicle, especially during ovulation. But there might also be some of those dynamics occurring throughout follicular genesis. I think this is really where, some of those human ovary atlas maps or looking at different proteins and gene expression profiles that exist across follicular genesis would really be helpful to see what type of matrosome modulating might happen throughout follicular genesis. But I think it's definitely both in some of those examples that I described about the fragmentation causing some primordial follicle activation and then of course the crosstalk.
And then from Ned Place we have, I can imagine your research, this is related to PCOS, sorry, I should read the full question. Your research on matrosome and degree of stiffness in postpubertal ovaries might have relevance or applicability to PCOS. So yes, go in the other direction.
Yes, I think I answered that question.
Are you specifically looking at helping PCOS patients as well, or is it more a bit of a side effect of what you're looking at in the normal ovaries?
Well, I think when you look at normal ovaries, I mean, I think the goal is to always identify what might be different or how disease modalities arise, because now you have this kind of baseline, concept to go after. And so I think that, looking at PCOS patients and collaborating with other people who do that might be really interesting and useful. Especially, I think that there's really, interstitial cell components that are also very interesting in PCOS, and so they might be secreting different matrix proteins or different signals. That would be interesting. And once we have those normals figured out, we could definitely, do those analyses.
Thank you. One last question, just because I think you've been grilled enough now. Is there any issue with the immunity if you're putting these scaffolds made from the matrosome proteins? Do you have any results on whether that affects, whether there's going to be an immune response to those, or is it?
Yeah, that's a great question. So that's definitely something we would have to test. So we are using bovine extracellular matrix protein for some of these scaffolds, which bovine, and we use bovine and porcine animals because we're able to get access to that tissue, but also because those are things that have been transplanted into humans before. Also, gelatin has an FDA approved use, and that was actually the first scaffold that we used. But that's something that we're going to have to test. But because we remove, I mean, there's still proteins there, of course, but which is the matrix, but because we remove all of the cellular materials, there should be at least less of an immune response. And the tissue that would be, or the cells that will be added back would be from the patient.
Yes.
Yeah.
Is there any possibility of creating those the scaffold from the human patient themselves, or is it just not enough tissue?
It would be really hard to do both of those things. What we might find is that there's a minimal working product of just, for instance, collagen 1, which you can buy commercially. You can get that as a plant-based source, and then some of these other matrosome components that might help appropriate follicular genesis at that rate that we want. So maybe we can create our own kind of matrix from off-the-shelf type products.
Great. All right, thank you very much. This has been really fascinating. I really enjoyed it. I don't think we've got any more questions in the Q&A. I think we just have a couple of housekeeping things. Yes, 1 minute. Great, right on time.
All right, perfect. And that is going to wrap up today's webinar. Thank you again, Jane and Monica, for a fantastic presentation. Just a quick reminder, you can find out more about this and other upcoming webinars by visiting www.ssr.org and clicking on the News and Events tab. Thank you all for your participation and enjoy the rest of your evening.
Thank you.
Salvo says thanks. Thank you, Monica. That was really fascinating.
Thank you, Jane. You were such a good moderator and you had such great questions. That was so fun.
Oh, well, because I was really fascinated as well. So I was trying to like put some of my questions in. That's great. Thanks very much. And yeah, enjoy the rest of your day.
Thank you. You too.
See you.